Welcome, everyone. I'm Naveed Sattar. I'm Professor of Metabolic Medicine at the University of Glasgow, so I'm a local boy. Cycled here, managed to fall off my bike, but I'm okay, thankfully. The unusual drizzle we have in Glasgow, which scuppered me. Anyway, this is, for those of you coming in, this is the session on accelerating global drug discovery, how UK Biobank is facilitating collaboration for drug development. I don't have too much by way of introduction other than I think you'll enjoy this. We have 6 talks, and then at the end of the talks we're going to have a very healthy question and answer session, so I would really encourage you to ask any questions you want. There are no such things as bad questions. They're all here to answer questions, anything you might wish to ask, and if not, I'll have lots up my sleeve. So let's start off with the boss, who's a good friend of mine, Naomi Allen. Naomi, come up, tell us about UK Biobank. Large, deep, long, and accessible. And it certainly is accessible because I think Glasgow, we've done very well out of UK Biobank and managed to publish over 100 papers since its inception just within our small unit. So, Naomi, let's kick us off.
Thanks very much. Gosh, these lights are blinding. So, welcome everybody. For those of you who don't know what UK Biobank is, I'm going to give an overview of this biological resource and how it can be used for discovery science, health-related research, and obviously drug development and targeting. Okay, so most people, when you mention the word UK Biobank, think of quite a large, whizzy freezer that just contains many millions of biological samples, and that's really the end of it. But actually, in reality, UK Biobank has a unique combination of size, containing 500,000 participants recruited throughout England, Wales, and Scotland, depth, in that we've collected a really large amount of information on all 500,000 participants, included very detailed data on genomics, circulating biomarkers, lifestyle factors, multimodal imaging, and so on, which I'll talk about in a bit later. It is a prospective study, so we have a long duration of follow-up. So when we recruited these individuals, we follow them up over time and find out what happens to their health through linkage to health-related records. And finally, and perhaps the most importantly, is the ease of accessibility to the data, with over 30,000 registered researchers globally now using UK Biobank to do health-related research that's in the public interest. So, just to put that into a bit of context, UK Biobank is a large prospective study. We recruited 500,000 individuals aged 40 to 69 years during 2006 to 2010. In England, Scotland, and Wales, we had an assessment centre here in Glasgow, actually, and we recruited by mailing out invitations to all individuals who lived within about a 30-mile radius of each of these 22 assessment locations. And we located the assessment centres in areas in order to maximise heterogeneity of the cohort. When participants came along to one of our assessment centres, we collected an awful lot of information about them, lifestyle factors, exposures to environmental hazards, personal medical history, and so on, a whole range of tests, including cognitive function, hearing tests, eye vision tests, blood pressure, lung function, body size, and we also collected biological samples, blood, urine, and in a large subset, saliva samples as well. Importantly, we obtained informed consent from all participants to follow their health over time through linkage to their health-related records, and also to recontact them in the future to do further data collection exercises. So when UK Biobank was set up, it was set up with the intention from the very offset of enabling research into the genetic and the lifestyle determinants of disease. So very early on, we knew that we wanted to perform genotyping on all individuals. And in 2017, we made available data from a custom-built genotype array with 850,000 variants, which was then imputed to over 90 million variants, and that was done for all 500,000 participants. Those data that were made publicly available in 2017 really led to a massive increase in the use of UK Biobank, led to hundreds of GWASs being performed. You can now see those results online on many search engines that will be done by the Broad, Stanford, also in Edinburgh. It's led to the development of polygenic risk scores, and that's been made possible through its large size, standardized assessment of all the exposures and health outcomes through electronic records. So it's hard to overestimate, actually, the impact that the release of these data has had on the research community, because it's been the largest study at that time in the world to release standardized genotyping data for such a large number of participants, together with all the lifestyle and the health outcome information. And it's been really transformative in enabling research into the genetic determinants of risk. So that closely led on to further work to do whole exome and whole genome sequencing, which other speakers in this session will talk in a bit more detail than me. But essentially, a pre-competitive consortium of industry partners came together to invest in UK Biobank to perform whole exome sequencing on all 500,000 participants. Data for the first 50,000 was made available in 2019. Data for the full cohort was then made available in the middle of last year. Follow on from that, a consortium public-private partnership of industry, charity and government came together to fund whole genome sequencing in all 500,000 participants. So if you cast your mind back, it was only 20 odd years ago that whole genome sequencing was done for one individual. Here we are, the technology's improved so much that we're now able to perform whole genome sequencing on 500,000 individuals within just a couple of years. Data for the first 200,000 participants was made available at the end of 2021, and data for the full cohort will be made available in November of this year. So I know the research community are waiting with bated breath for those data. But these types of consortium that are using UK Biobank in this way really does illustrate the power of open science, collaborative science, multidisciplinary science, and pre-competitive industry collaboration. And it's just been fantastic to be able to see industry and government coming together to really fuel science and make those data publicly available for researchers around the world to use. So in addition to the genomics data, we've also focused on performing other cohort-wide sample assays, including a range of biochemistry markers, hematology markers, telomere length, which is a proxy marker of biological aging, circulating metabolites performed by Nightingale, which is mainly lipids, circulating lipids. And in addition to that, we've also performed some pilot work, both on the transcriptomics, doing single-cell RNA sequencing on 5,000 individuals, proteomics, which I know other speakers will talk about in more detail, performed by the Olink platform in over 50,000 individuals, and the metabolomics, as I've said. And this increasing momentum of these pilot studies will, in the future, I strongly suspect, will lead to cohort-wide scale assays of doing RNA sequencing, targeted and untargeted mass spec for both metabolomics and proteins. And I also suspect that there's also a lot of interest in performing cohort-wide assays on markers of infection, markers of the immune system, epigenetics, and so on. And the rate that the technology is changing in being able to perform high-throughput, cost-effective assays in this way for population health research is really exciting. And there's simply no way, even 3 years ago, we would have imagined that this would even be possible. So in addition to the sample assays, we're also characterizing participants through an additional study to perform multimodal RNA MRI scans. So we aim to perform multimodal imaging on 100,000 participants by the end of next year. We've already done 70,000 participants. They've come to one of 4 assessment centres around the UK. We do a brain MRI, heart MRI, and an abdominal MRI, plus a full body DEXA scan of the bones and joints, and a carotid ultrasound, together with a repeat of all the measures that were performed at baseline. And I have to say, this is by far, by some margin, the largest study in the world to have multimodal imaging data together with the genetic data, the lifestyle, and the health outcomes to be able to facilitate research into the underlying pathology of early disease. And we're now just started a repeat imaging study to bring back 60,000 of the original 100,000 back for a second scan a couple of years after they've had their first scan, in order to enable research into changes in internal organs over time and how that relates to disease risk. And that also includes a full repeat of the baseline measures plus some eye measures as well. And this is really important for the research community because for the first time at scale, it will enable research into how genetics and lifestyle factors influence the internal pathophysiology prior to clinical diagnosis. So there's no other study in the world quite like it. So as I've said, UK Biobank's a prospective study. We follow up participants over time to find out what happens to their health through linkage to electronic health records. We now have 15 years of complete follow-up to national death and cancer registries, hospital inpatient records, including critical care, COVID testing data, and for just under half the cohort, primary care data, and efforts are on way to increase that coverage. As you can see, by the end of 2020, we already have thousands of incident cases of the most common diseases, and over the next 5 to 10 years, you can see that the numbers of new cases of disease will really enable robust research into the genetic and lifestyle determinants. So finally, I just want to talk about the ease of accessibility of the data, and I know that another speaker in this session will talk a bit more about mechanisms through which the data can be accessed. So right from the offset, UK Biobank was set up to be easily accessible by both academic and commercial researchers on exactly the same basis worldwide. We now have over 30,000 researchers. You can see the number of researchers is just increasing exponentially, particularly since the release of the genotyping data in 2017. We have researchers from all over the world, and we now have 7,000 publications coming from the resource, 2,000 of which were from last year alone. So lots of researchers are starting now to use UK Biobank from around the world and are recognizing what a unique resource it is. So, you know, the reason for making it so easy to access the data is that it maximizes the usage of the resource by the global research community to make scientific discoveries. It enables collaborative research, and of course it enables researchers to refute or build on or share each other's work in a truly open forum. So finally, I don't want to talk too much about this because I know Asha will speak about this later in this session, but in 2021, we launched our research analysis platform, which is a cloud-based, secure platform whereby researchers can log in and do their analyses in a secure way online rather than having to download the data. And this is really starting to help democratize access because it brings the researcher to the data rather than the data to the researcher. It means researchers only need an internet connection, so researchers from low middle income countries can access the data in just the same way as a researcher from another institution. And AWS have also made free research credits available to researchers from low middle income countries and all the early career researchers as well. So if you are an early career researcher, I strongly recommend that you have a look at the website, and if you want to access the data, you can do so through the research analysis platform because AWS are effectively making it free to do so. So finally, just to summarize, UK Biobank is a pretty unique resource, and that's because of its combination of at least 4 dimensions: the scale, over 500,000 participants, the depth of data that we have on almost all of our participants, the duration with 15 years of complete follow-up to health records, and the ease of accessibility of the resource. So I strongly encourage you to have a look at the website, find a bit more about it, and see if it will be useful for your types of research. And for sure, the best is yet to come, so keep a lookout for UK Biobank. So I'd just like to acknowledge the UK Biobank team, our funders, and most especially of all of our 500,000 participants. Thank you very much.
Thanks, Naomi. I think we're having questions at the very end. I think that was the plan. Yeah. So let's move on to Abhishek Nag, Dr. Abhishek Nag, who's going to talk about novel obesity targets with exome sequencing data. And this is kind of exciting. I'm involved in some of the ongoing trials with incretins and dual and triple agonists with obesity, which is really exciting. I'm also helping the UK government as part of the obesity initiative. So this is one of the biggest tasks, biggest areas of research, I would suggest. So over to you, Abhishek.
Great. Good morning, everyone. And firstly, I would like to thank the organizers for providing me the opportunity to present our work over here today. So as was introduced, I'm going to provide an overview of how large-scale human exome sequencing data is being harnessed currently and the recent advances that have taken place in identifying drug targets. And in particular, I'm going to talk about how this approach has been applied in the UK Biobank cohort. So that leads me to my first slide. So as the speaker before already spoke about this in detail, and I'm sure most of you are quite familiar with the UK Biobank, so I won't spend much time on this. So as you know, UK Biobank is a large biomedical database and health resource of about 500,000 individuals from the UK Biobank that were aged between 40 and 69 years at the time of recruitment. Now, there's a vast array of health information that is available in the UK Biobank cohort, and this includes self-reported diagnosis, hospital and death records, anthropometric data, blood and urinary clinical biomarkers, and most recently, even imaging data has become available. Now, in terms of genetic data, there's also various forms of that have become available in the UK Biobank over the years. And this includes microarray data, which mainly captures common and low-frequency variants that is found in the population. We have also become available with whole exome sequencing data in the UK Biobank, which captures sequence information in the coding regions of the genome. And more recently, as was described by the speaker before, whole genome sequencing data has also become available in the UK Biobank. So as I mentioned, the focus of my talk is going to be around the applications of whole exome sequencing data in this cohort. So what's the value of having whole exome sequencing data in the UK Biobank? So firstly, it provides us with a catalog of rare to common coding variants that is found across the human exome. And in the UK Biobank, we were able to identify approximately 2.7 million such coding variants. Secondly, exome sequencing can also help us identify what we call human knockouts. Now, what I mean by human knockouts are individuals in the population that have a loss of function or protein truncating variant that affects both copies of their gene. So one would expect that such individuals will have no functional copies of that given gene. And such human knockouts are valuable for drug discovery because they serve as in vivo models to study the systemic inhibition of gene products. So the figure on the right over here shows the genes for which we observe complete human knockouts in our database. So the plot in yellow describes the data for UK Biobank. And now, with the inclusion of additional sequences in our database, which is shown by the blue curve over here, you can see that in the combined data set, which includes the UK Biobank and the additional sequences, we have a greater number of genes for which we observe complete human knockouts. So just a slide on the UK Biobank phenome. So we analyze a diverse range of phenotypes in our whole exome sequencing analysis. And this includes over 1,400 quantitative traits, which includes some clinically relevant biomarkers, over 15,000 clinical endpoints, which includes some clinically relevant union phenotypes that we defined for our purposes. And more recently, multi-omics measurements have also become available in the UK Biobank, and this includes the Olink proteins and the Nightingale assay metabolites. So now coming to the applications of whole exome sequencing to the drug development pipeline. So it does have applications throughout the drug development pipeline. So the first and the most important one of which is for target identification and validation. Now, it has been shown that human genetics validated targets are actually 2 to 3 times more likely to lead to approved drugs. Secondly, whole exome sequencing also has application in the precision medicine strategy by identifying patient segments that are most likely to benefit from a treatment. And thirdly, it also has application in drug safety assessment. So one can do phenome-wide scans, which can help target any safety signals that might be associated with the target of interest. So I'm going to talk a bit more about the first application, which is around target identification using whole exome sequencing data. So the cornerstone of using whole exome sequencing data for target identification is actually identifying gene-phenotype relationship. Now, as you must be knowing, that previously genome-wide association studies have identified several common variant loci that are associated with human phenotypes. But a challenge with GWAS associations was that they were actually limited in terms of their translational potential. So now, with the availability of whole exome sequencing data, what we are able to do is test rare coding variants for association with human phenotypes using a framework, what we call PheWAS, or phenome-wide association study, which can help identify novel gene-phenotype relationships which are driven by these rare variants. And what's the benefit of these rare variant-driven gene-phenotype relationships, especially the protective associations, is they can help us identify potential therapeutic targets. And I'll show you that later on in one of the examples, how we can actually identify such protective associations. So I mentioned in the previous slide about performing a PheWAS using sequencing data. So how is a PheWAS performed? So we used an approach what's called collapsing analysis to perform our PheWAS. Now, briefly, what a PheWAS involves is for each of the approximately 19,000 genes that are found in the human exome, we test the aggregate effect of rare functional variants, one gene at a time. And we do that using different collapsing models. Now, put simply, what these collapsing models differ in are the filters that are used for variant selection. So we then test the aggregate effect of rare variants in each gene for all the clinical endpoints and quantitative traits that we have access to that I described previously. And this led to a whopping 3.4 billion association tests that we performed in our PheWAS. So what were the findings of our PheWAS? Now, in the world of genetics, as some of you might be knowing, we tend to use a very stringent threshold for significance, and that's because of the large number of tests that we perform. So using a p-value threshold of p less than 1 into 10 to the power minus 8, we identified 1,732 significant gene clinical endpoint relationships in our PheWAS, and similarly, we identified close to 1,500 significant gene quantitative trait relationships. Now, what were these significant findings? Now, just a brief summary over here is that it was reassuring to see that we identified some very well-established gene-phenotype relationships that have been reported in the literature, including association between variants in BRCA1 and BRCA2 with breast cancer, HNF1 alpha and diabetes, PKD1 and PKD2 with chronic kidney disease. But we also identified some potentially novel and understudied gene-phenotype relationships. Now, we have made available all the association statistics of our PheWAS analysis through our publicly available PheWAS portal, the link to which is at the bottom of this slide. So it's not just us, but several groups across the world that have actually tapped into this opportunity of analyzing the large-scale exome sequencing data that has become available in UK Biobank and some of the other population-based cohorts, which has led to the identification of several very important protective gene-phenotype relationships for several human traits. and have provided some high-impact publications of those over here. So these include the association between CDEB and chronic liver disease, MAP3K15 and diabetes, INHBE and abdominal obesity, GPR75 and obesity, and HSD17 beta 13 and chronic liver disease. Now these are just some of the examples. Obviously it's not an exhaustive list. So I'm going to talk a bit more about the protective association between loss of function of MAP3K15 and diabetes, because that was identified by our group. So the association between MAP3K15 loss of function and diabetes actually provides an exemplar of a protective gene phenotype association that was identified in the UK Biobank. So what we observed is that recessive carriers of loss of function and missense variants in this gene MAP3K15 had reduced odds of developing diabetes. And this association was statistically significant, as evidenced by the p-value of 5 × 10⁻⁹. Now, we next tested only the recessive loss of function variant carriers in MAP3K15, and we observed that those individuals had about 35% reduced odds of developing diabetes. Now, consistent with the protective effect on diabetes, the MAP3K15 variant carriers also had significantly lower HbA1c levels, both when testing the recessive loss of function and missense variants, and separately when only testing the recessive loss of function variants. We were then able to replicate our finding in two genetically diverse cohorts, namely the Mexico City project and the FinGen Biobank. Now, in the Mexico City project, we were able to replicate our association between MAP3K15 and self-reported diabetes, while in the FinGen cohort, we were able to actually replicate the protective effect of MAP3K15 loss with both clinical type 2 diabetes as well as type 1 diabetes. And the latter, we feel, is likely due to the higher prevalence of type 1 diabetes in the Finnish population, which provided us with adequate power to detect the protective association with type 1 diabetes. So there are a few aspects about the association between MAP3K15 loss of function and diabetes, which have clinical impact that I would like to highlight. So the first of which is that we observed that the protective effect of MAP3K15 loss and diabetes is actually independent of BMI. So we performed the same analysis that I showed before, now adjusted for BMI, and you can see that the protective effect is pretty much consistent with what was observed previously. So this suggests that the underlying protective mechanism of MAP3K15 loss of function with diabetes is likely independent of BMI, and therefore it might be mediated via insulin resistance independent pathways. Second, we also observed that the protective effect of MAP3K15 on diabetes as well as the biomarker HbA1c amongst heterozygous female carriers whom we expect to have 50% loss of the gene was less as compared to hemizygous male carriers whom we expect to have 100% loss of the gene. And this indicates that the effect of inhibiting this target might be dose dependent. And lastly, as I had mentioned before, that we can also look for any safety signals and try to de-risk a target using human genetics data. So we did the same for MAP3K15, where we performed a phenome-wide scan of MAP3K15 loss of function across all the 15,000 clinical endpoints that we had access to in the UK Biobank, and we did not see any safety signals, even at a suggestive p-value of 10⁻⁵. So that brings me to my summary slide, which is, in the recent years, large-scale whole exome sequencing has become possible, including for population-scale biobanks. And whole exome sequencing has important applications throughout the drug development pipeline, including for identifying novel drug targets. And hopefully I was able to demonstrate that identifying loss-of-function variants that protect against disease can provide human genetics validated therapeutic targets. And lastly, the acknowledgements. So I would like to thank the contribution of my colleagues at the Center for Genomics Research at AstraZeneca, and obviously to all the volunteers and participants of the UK Biobank, without whom all this work wouldn't have been possible. Thank you very much.
Thank you very much. That was a lovely talk. So let's move quickly on. We're actually bang on time. We're okay. So Adrian Cortes, who's from GSK, continuing sort of drug discovery theme, Adrian, I gather. global collaboration and use of UK Biobank for drug discovery. Over to you.
Thank you. Okay. Good slides. Great, thank you. Well, thanks a lot for the invitation to come speak to you today. I was suggested this title when I was invited, and I kept it because I wanted to reflect on three items in this title. So global collaborations, UK Biobank, and drug discovery. So I will talk about genetics in drug discovery, why UK Biobank resource for drug discovery, why global collaborations, and then talk about some of the work that we've done in. Oh, okay, sorry, thanks. Okay, good. Okay, so probably you've seen this graph in the past, and it's daunting probability of success of assets moving through clinical development in the pharmaceutical industry. And here you can see overall probability of success of less than 20% for phase 1 assets, right? And costing the industry a lot of money. And if we look at why are these targets failing, it's mainly because we're hitting the wrong targets, right? So efficacy is the main explainer here. So how can we work on identifying targets early that will maximize efficacy? So this is where genetics comes in. And this is a graph from a paper published by colleagues at GSK about 10 years ago, where they looked at clinical trial data from the past decade, and they looked at, for each of these targets, which had genetic evidence and which ones didn't, right? And they made this observation that targets with genetic evidence basically had a high probability of success across all phases of clinical development. So this accelerated the adoption of genetics for drug discovery. And in principle, you can hypothesize this very simply, is identify genetic variants that instrument gene function, and we saw really nice examples in the previous talk, and then look at whether these genetic variants affect risk to disease, right? So, and then hypothesize this as a therapeutic target for this disease. And GSK made this strategic decision to be driven by genetics a few years ago. So why genetics as a starting point for drug target hypotheses? And already this was alluded to, genetics is a very powerful tool. We can think of them as experiments of natures, right? So genetic variants are naturally occurring in the population, and then we can observe this genetic variation and then look at their effects in disease outcomes. There is no reverse causality, so alleles are fixed at birth, so we don't need to worry much about confounding. And it's a powerful causal link from gene to disease. So it not only allows us to do target discovery, but also development. So it allows us to predict efficacy, on-target effects of target perturbation, so for clinical, for indication expansion, or for potential adverse effects of on-target perturbation. So we already saw a summary of UK Biobank twice, so I will just focus on some of the aspects that I find very important. So one is scale and access of the data. So previous to UK Biobank, access to data was difficult, or a major challenge, right? And UK Biobank brought a step change to data access, not just access, but also scale. And this data was made available to academia, industry, and made available globally. And we already looked at some of the many different modalities that UK Biobank is measuring across health-related outcomes by linking health registries, genetics, first the array-based data, now whole exome and whole genome sequencing, molecular phenotyping, and here I'm just highlighting a few that we use routinely, so proteomics, metabolomics, serology measurements for antigens from infectious diseases, and blood and urine biomarkers, and MRI imaging. And I highlighted in blue those that have been funded through public-private partnerships. So again, coming back to this global collaboration aspect. So I wanted to give you an anecdote of just to demonstrate how UK Biobank can accelerate drug discovery. So GLPR, so the receptor, encodes for the receptor for this hormone stimulates insulin release, right? And we've seen a lot of this in the media recently. And targets for GLPR are effective therapies for type 2 diabetes. And in 2008, the FDA announced that any new type 2 diabetes drugs should not have an unacceptable increase of cardiovascular disease risk. So we can look at genetic variants that instrument the effect of modulating GLPR1. And this is work done by my colleague Robert Scott and at GSK. And when he was in Cambridge, 2016, he looked at the genetic effects of instrumental genetic variants or GLP1R in type 2 diabetes and other relevant disease outcomes. And you can see here the protective effect of the particular genetic variant in type 2 diabetes and cardiovascular disease. And what I want to highlight is not necessarily the different indications shown in this graph, but just how long it took back then to perform this analysis, right? So he tells me that it took about 18 months, thousands of emails, hours in calls. UK Biobank comes in a couple of years later. Same analysis, now will take him 3 hours and 3 emails. And you can see how this accelerates our ability to build therapeutic hypotheses. And you just don't want to do this for a single genetic variant, but you wanted to do it for every single genetic variant that you observe in the cohort for every single clinical endpoint that you have access to. And that's what we routinely do now. So why global collaborations? Well, I think that this enables the enrichment of the UK Biobank through these public-private partnerships. So it's an incremental build of this very deep data, as we heard back. So expanding different data modalities. Why would industry engage in this? Well, I don't think that us alone can actually do this, right? And there's many reasons why that is: financially, operational, sample availability. So engaging with UK Biobank is a way for us to enable this science and get access to it as well. And of course, then bring further potential for this resource to accelerate identification of drug targets and then the development of medicines. And we already saw how the exponential growth of scientific publications, just demonstrating how this resource enables basic science. So 2 different public-private partnerships that I will just highlight in the context of UK Biobank. So we will hear later from Chris on proteomics, so I will not steal his thunder. But this consortium was set up to fund the generation of proteomics data using the Olink platform for measuring protein abundance for 3,000 proteins in 50,000 UK Biobank participants. And I'm pretty sure we're going to hear more about it later. And then also mentioned the UK Biobank whole genome sequencing project. So this is a public-private partnership between 4 pharmaceutical companies, 2 public agencies, and sequencing at 2 different centers, so the Wellcome Sanger and Deco Genetics. And what I want to highlight here is, sequencing started before actually COVID. Data has finished sequencing, and the 4 funding partners have actually received the data early this year. It was an enormous effort to get this data through, and this is a great admiration for colleagues at Sanger and Deco that performed this despite major disruption to supply chains during the COVID pandemic. So why would you want to generate the whole genome sequence for UK Biobank participants? Well, this gets us closer to completion of participants' genomes, an extensive characterization of sequence diversity in the population. It improves measurement or observe variants at low frequency spectrum, absent from array genotyping and imputation. You don't need to select the parts of the genome that you will sequence like you do in whole exome sequencing. So even if you look at the exonic variants captured by the whole genome sequence, you gain about 10% of the variants absent in the capture arrays, and better identification of more complex genetic variants, such as structural variation. So I will just walk through how then you can scale this analysis and what type of inference you can read from this data. So now we're talking about how do you integrate all of these different data sets in UK Biobank for drug discovery, right? So I mentioned proteomics data, so we have abundant measurements for 3,000 proteins. If you look at the genome and you look at genetic variation correlates with protein abundance, you get this type of figures here called Manhattan plots, where each peak is a genomic region associated with protein abundance. So most of these proteins contain QTLs, quantitative trait loci. And then we can do a test called colocalization with disease data, which basically aims to test the hypothesis that a genetic variant that is disease-associated is shared with a protein, with a variant that causes changes in protein abundance, so causal sharing of variants. And when you do this type of test with data from for hundreds of disease outcomes, you generate basic hypotheses of target indication pairs, right? So in this case, we generate about 2,000 colocalizations for PQTLs. Now, when you look at this data, then you observe that actually, well, there's actually not a single genetic variant that instruments gene function or protein function. Even within a genomic region where you encode the gene, there's multiple distinct genetic variants that could be affecting protein abundance. So you can perform conditional analysis, and then you can start performing this colocalization analysis, not just with the single most significant genetic variant, but all of the conditionally independent variants that you observe in this analysis. And this allows us to improve our ability to overlap GWAS data for disease and proteomics data. And what this allows us to do is to then start building what we call this allelic series across the allele frequency spectrum of different types of genetic perturbations that modulate or alter protein abundance, but also related phenotypes. So, for example, here I have PCSK9 and adipolyprotein B, and different types of genetic variants that are conditionally independent. You have orange. These are mainly common variants, what we call here the colocalization ones. Oh, sorry. Then you have loss of function variants in green, and missense variants in blue. So what you can see is that variants with very strong effects are more likely the ones with the low frequency spectrum. Variants with low effect are more common, so the ones in orange. But they all have this relationship between the effect of gene protein function and then the measured phenotype on the y-axis. And on the right-hand side, we do similar analysis, but now with MEP levels and bone mineral density. So we believe that this allelic series then strengthens our ability to make very strong gene to phenotype connections. Okay, so now another thing that we can do is that we already heard about region-based tests or burden tests, right? So the idea here is, can you aggregate genetic variants that are very low frequency in the population into a single test to increase your statistical power? And one of the difficulties here is to actually, how do you annotate a genetic variant that, for this type of aggregation? So you can use in silico models to predict what is a protein loss in function or missense variants. And that's all like what we call a conventional way. But then you can integrate the proteomics data to use the assay data to actually annotate the genetic variants. And what I want to show you here is that on that forest plot here, the red tests are the ones that are like aggregation of variants that are enhanced with the proteomics data relative to the blue ones, which are just the in silico annotation of genetic variants. And you can see how you can increase your power on the test by using this functional data for annotating and aggregating genetic variants for the burden tests. So I will end there, and just final remarks. So I think that these global collaborations have enabled and enriched the UK Biobank and generated data at an unprecedented scale. Supports the development of UK genome resources and improves our ability for using genetics for drug discovery. And I would like to thank collaborators at GSK, colleagues in the UK Biobank Whole Genome Sequencing Consortium, and all UK Biobank participants. Thank you very much.
Thank you, Adrian. I enjoyed that very much. I was involved in that Robert Scott paper, so it's nice to see that up there. So we're halfway through. We've got 3 more talks. So Helen, come up. So, sorry, Mary Helen Black, who's from, I think, from Philadelphia, if I picked up correctly, or is from Foresite Labs, Whole Genome Sequencing for Drug Discovery. And whilst Mary Helen is coming up, please think of the questions you would like to ask of our panel, because we have plenty of time for questions. They're all on time. So any question is, and that you've always thought about, please to ask. You've got a fantastic panel to ask. So Mary Helen, over to you.
Thank you very much. And I'd like to thank the organizers for having me here today. From Foresite Labs, which is actually in Boston, although I am located in Philly, on leveraging the UK Biobank to enable precision medicine-focused innovation in biotech. Foresite Labs is a biotech incubator, and so you can imagine there are multiple ways that these data are incredibly useful to us. Some disclosures, and you've already heard Naomi and several other speakers describe the vast constellation of data available in the UK Biobank, and I just want to emphasize its uniqueness and the value of UK Biobank's open access policy, and really the continued need for additional molecular data to drive advancements in biomedical research. So the UK Biobank is the largest comprehensive source of individual level data accessible to researchers today, and at a modest cost. The data not only include a wide variety of phenotype information from hospital and primary care records, interviews, targeted questionnaires, biomarker assays, and multimodal imaging studies, but also supply a growing set of molecular omics data that has been used to inform drug discovery and development, as you've already heard the other speakers describe. So how are these data leveraged for therapeutics discovery? There's several ways that pharma and biotech think about using all these data, and many use cases. So for example, novel drug target discovery using genetic variation as a proxy for pharmacologic modulation, which colleagues have already described, particularly for obesity. Prediction of adverse events that may occur in the treated human population, which can help inform safety and guide mitigation strategies. Assessment of opportunities for drug repurposing through analysis of pharmacy exposures and genetic variants underlying drug targets. And examination of patient stratification to better understand patient heterogeneity and improve response to therapy. So for the purposes of my talk, I'll mainly focus on examples from drug target identification and patient stratification in the pursuit of precision medicine strategies that maximize efficiency in patients most likely to benefit from therapy. I actually just have one example here, and you've heard several from the other speakers as well, on novel drug targets that have been identified from UK Biobank whole exome and whole genome sequencing. This example here is actually from a study conducted by Alnylam. So Alnylam authors examined data from UK Biobank participants with whole exome sequencing, and at the time restricted analysis to unrelated individuals of Northern European ancestry from the UK Biobank. They identified a signal in the inhibin subunit beta E gene, or INHBE, which encodes a secreted protein primarily expressed in the liver, and is associated with abdominal adiposity as measured by waist-hip ratio adjusted for BMI. The variants identified were predicted loss of function variants and were associated with reduced adiposity and lower incidence of type 2 diabetes and coronary heart disease, suggesting pharmacologic inhibition of INHBE may be a plausible treatment for obesity and cardiometabolic disease. And then after replicating the associations in other human cohorts, the investigators functionally characterized the variants in vitro and found that decreased gene and protein expression led to healthier fat distribution, whereas elevated levels were associated with obesity and insulin resistance. Now, based on these findings, Alnylam initiated a program, currently still in early testing, to develop an antisense oligo targeting INHBE as a novel drug target for obesity, which they propose could also serve to complement existing therapies for diabetes and cardiovascular disease. So it's been widely reported that modulation of drug targets in specific disease indications that are supported by human genetic evidence are more than twice as likely to transition from phase 1 trials to full approval than drugs without that support. Given the wealth of population-based genomic data linked to clinical information that's available today, it might not be surprising that 2/3 of the 50 FDA-approved drugs in 2021 were supported by genetic evidence, as shown in a paper by Ochoa et al. Interestingly, only about 12% of those drugs approved included a precision medicine strategy, most of which were cancer treatments approved for patients with specific mutations. And interestingly, among the manufacturers of those 50 novel therapies, 64% were developed by small or early biopharma or biotech companies, most with less than $1 billion in annual revenue, but leading drug innovation. And reviewing FDA approvals in the past year and a half to date and applying a similar methodology to Ochoa et al., we found that 73% of drugs approved in 2022 and about the same proportion in 2023 were also supported by genetic evidence. So I'm going to walk through a couple of use cases now, sort of focusing on precision medicine strategies. An example of a recently approved drug actually also comes from treatment of autoimmune disease. Last September, BMS's oral TYK2 inhibitor was approved for treatment of moderate to severe psoriasis. It's well known that the JAK-STAT pathway regulates intracellular signaling of cytokines in IL-23, IL-17 axis that are central to psoriasis pathogenesis. TYK2, a key member of this pathway, has been considered a susceptibility locus for psoriasis for more than a decade, with the first GWAS pointing to a signal for TYK2 actually in 2010. But in 2017, a large meta-analysis of exome data in nearly 12,000 psoriasis cases and 29,000 controls identified rare and low-frequency functional variants in TYK2 that were protective for psoriasis and can be considered pharmacomimetic for TYK2 inhibition. The POETIC trials demonstrated efficacy and safety of TYK2 for treating psoriasis. It's fairly well tolerated with few serious adverse events, although the highest response rate for any endpoint was only 58%. So psoriasis, like most autoimmune diseases, is complex, and the affected patient population is diverse. Accounting for this diversity among patients is essential for development of better therapeutics and identification of patients who are most likely to respond to these therapies. Now, to that end, analyses of UK Biobank data can help elucidate those characteristics. So, for example, a recent analysis of UK Biobank genetic and lifestyle data found that psoriasis polygenic risk score and unhealthy behaviors, such as smoking, diet, and physical activity, were predictive of incident psoriasis. Overall, high genetic risk score alone increased psoriasis risk by 34% compared to those with low genetic risk. And among individuals with poor lifestyle characteristics, high genetic risk score nearly doubled psoriasis risk. Put another way, as shown here on the bottom right, the joint effect of poor lifestyle and high genetic risk was greater than 4-fold risk of psoriasis relative to those with low genetic risk and healthy lifestyle. So this information may be useful in consideration of clinical trial development and precision strategies for new therapeutics. And as sort of proof of this concept, Prometheus Biosciences recently announced results from their Artemis UC and Apollo CD phase 2 trials of anti-TL1A monoclonal antibody in the treatment of ulcerative colitis and Crohn's disease. So TL1A is encoded by the TNF-SF15 gene, a well-known locus for inflammatory bowel disease, or IBD. Leveraging the knowledge of genetic variants reported for association with IBD in the literature and studies conducted in collaboration with Cedars-Sinai, Prometheus developed a polygenic risk score for TL1A expression or activity, and then planned to implement that risk score as a companion diagnostic for anti-TL1A therapy. The Artemis UC trial design actually consists of 2 cohorts, one cohort for evaluation of efficacy and one cohort enriched for their companion diagnostic positive patients. Interim analysis from cohort 1 has been recently presented, although full phase 2 results are not yet available. So far, with roughly 60 to 70 patients in each of the treatment and placebo arms, primary endpoints of clinical remission and secondary endpoints such as endoscopic improvement were observed at week 12 of the study. Now, importantly, even with a modest number of patients in this interim analysis, Prometheus was able to show that roughly 38% of patients positive for the companion diagnostic based on a genetic risk score were more likely to benefit from therapy compared to 25% of all comers, as shown here in the top right figure. While the Apollo CD trial design was constructed a bit differently, with a single cohort evaluated at 12 weeks followed by an open label extension study, similar interim positive results were observed in terms of efficacy and safety. And again, even with a modest number of trial enrollees, 57% of companion diagnostic positive patients experienced clinical remission compared to 49% of all comers, as well as 45% versus 26% with endoscopic improvement, as shown here in the bottom right figure. So another use case comes from studies of migraine, one of many common complex neurological disorders. In March of this year, FDA approved Pfizer's Zavzpret, a calcitonin gene-related peptide, or CGRP, receptor antagonist nasal spray for migraine treatment. Several CGRP receptor antagonists have actually been developed in the past few years, either as injectable monoclonal antibody or oral small molecule therapies. And indeed, CGRP is known to be a major component in migraine pathogenesis and has been a focus of migraine research since the 1980s. CGRP receptors are expressed centrally and peripherally throughout the nervous system and act as cerebral vasodilators. Antagonists to these receptors increase inhibitory mechanisms to desensitize neuronal circuits before or during an attack. 2 recent large-scale meta-analyses of UK Biobank and other cohort genotype data yielded significant signals in calc-A and calc-B, as shown in the locus zoom plot here on the right. Calc-A and calc-B jointly encode CGRP, and the calc-A, calc-B variants identified in the GWAS also have cis-EQTL effects and are supported by co-localization evidence consistent with upregulation of CGRP in migraine cases. Several trials for CGRP antagonists have been conducted, but the highest response rate for any endpoint is roughly 40%. So migraine is such a heterogeneous disorder affected by many factors, including smoking, alcohol intake, diet, low vitamin D, comorbid conditions, and genetic variation. And in the past 5 years, nearly 36 studies of migraine have been published from UK Biobank data, highlighting a role for each of these factors in this disorder. So polygenic risk scores have also been developed for migraine and shown to be predictive. And in a recent paper by Coleman et al., have demonstrated utility in identifying responders to migraine medication. So shown here in the plot on the bottom right, the authors scaled the PRS effect to a 2-fold increased risk of migraine. So what this plot is showing you is that a 2-fold increase in migraine risk, represented by the polygenic risk score, was associated with positive response to migraine-specific treatment, particularly for triptan therapies. And while the study didn't evaluate CGRP receptor antagonists, it demonstrates evidence of treatment effects that may vary by patient's genetic architecture. So interestingly, Amgen completed a phase 4 open label study of irenomab, which is an antibody treatment, to assess whether response to irenomab varies by high or low genetic risk for migraine. Results of that study are not yet posted, so whether such precision medicine strategy will work for those agents is yet to be elucidated. But again, the Kogelman study suggests genetic architecture may modulate treatment efficacy. So in summary, large-scale population-based resources such as the UK Biobank can be leveraged for a wide variety of purposes in pharmaceutical development, including to provide genetic support for emerging therapies, as well as identify novel targets for new medicines, and to better understand patient heterogeneity in the pursuit of precision medicine strategies to improve response to therapy in those who can benefit most. Nearly 3/4 of novel drugs approved by FDA are now supported by genetic evidence, yet only a few, typically oncology-specific therapies, include a precision medicine approach to treatment. So use cases for assessing patient stratification to identify subgroups who will most benefit from therapy are beginning to emerge in common complex disease, especially those with high unmet need. And they represent an opportunity for much needed healthcare innovation. Thank you.
Excellent, Mary Helen. We'll move the last two talks. Now we're going to shift to proteomics. And Chris Whelan, who's also come from the US, I think, and is currently seeing family in Dublin. So Chris, tell us about the value of large-scale proteomics for understanding mechanisms. And as before Chris starts, again, remember questions. So we have 2 more talks, and I want lots of questions. Otherwise, I will be dominating all the questions. I have at least 10 for each speaker, potentially. I'm only joking. But I've got a few. So Chris, over to you.
Fantastic. Thank you very much. So you've already heard from Dr. Black, Dr. Cortes, and Dr. Nag about how UK Biobank is enabling precision medicine in drug discovery. And my first slide is just going to reiterate that. You've heard this a few times already, but UK Biobank is essentially the cornerstone of everything we do as geneticists in pharma R&D. We're using UK Biobank to find associations between genes and diseases using GWAS, rare variant burden testing, and XWAS. And then we want to turn those associations into new drug programs for the reasons that you've already seen and already heard a few times. Drugs developed with supporting evidence from human genetics are at least twice as likely to be approved. So genetics holds a lot of promise as sort of a Swiss Army knife for R&D in pharma, but it's not a perfect tool. Oftentimes, it will point towards gene regions associated with diseases without necessarily giving us the causal gene. And in those instances where we can find the causal gene, the underlying mechanisms are not entirely clear. So this has prompted us to maybe consider revising this G2D2 genetics-guided drug discovery model and incorporate proteomics part and parcel of the process. So what do I mean by proteomics? Well, simply put, it's the systematic measurement of many different proteins at the same time. And why proteins? Well, first of all, obviously proteins represent the majority of our drug targets, and they also represent quite a few biomarkers. There's a number of established markers like ApoB for familial hypercholesterolemia, and there's also a number of new markers that, for example, Janssen is using in our clinical trials, p-tau217, which is a blood-based measurement, essentially giving us a measurement of brain amyloid for Alzheimer's disease trials. Now, what's notable is that the field of proteomics has really exploded over the last 10 years or so. Traditionally, if you wanted to measure many different proteins at the same time, you'd use mass spec. Now, mass spectrometry is still the gold standard in many ways for multiplex proteomics, but more recent developments have allowed affinity-based technologies to enable multiplex proteomics measurements at higher throughput and using much smaller sample volumes. And this has been recognized. These recent technological advances have been recognized by industry leaders in the field of genomics. Carsten Surrey, Jochen Schwenk, and Mark McCarthy, for example, recently commented on how proteomics is starting to take its place alongside genomics for the comprehensive characterization of human health and disease. So recognizing the transformative potential of proteomics, we came together as 13 different pharmaceutical companies and formed a consortium called the Pharma Proteomics Project. The purpose of that consortium was to generate just under 3,000 protein measurements from the blood samples of over 54,000 people in the UK Biobank. And the overall goal here was to accelerate the identification of better genetic drug targets, better biomarkers, and more efficacious medicines. So briefly, we used a technology developed by Olink Proteomics in Uppsala, Sweden. Very simple explanation of this technology is that it relies upon matched pairs of antibodies. The antibodies must be in close proximity. They must be essentially binding the same antigen. If they do that, they hybridize, and that provides a unique template for DNA polymerase-dependent extension. They get extended into amplicons, and then those amplicons can be read by a next-generation sequencer like a NovaSeq as a protein measurement. And so we applied this Olink technology across 54,219 UK Biobank participants. Of those 54,000 or so participants, most were randomly selected. This was intentional. We wanted to preserve the overall structure of UK Biobank as much as possible. There is a small number of pre-selected samples, about 6,000 pre-selected samples, as well as a small number of participants from the COVID-19 imaging sub-study that took place during the height of the pandemic. And as you'll see from the bottom left, in blue, the randomized cohort looks basically the same as the broader UKB cohort. The pre-selected samples in red, as you might expect, are enriched for some diseases. And this is just to say that this was a pretty efficient study. We started it in April of 2021, and the first batch of 1,500 proteins was generated by December of that year. Now, we did double the number of proteins. It's now 3,000 proteins, and we are currently working with those data. The first 1,500 are now available to the community, and the second 1,500 will become available in the fourth quarter of this year. And this represents the world's largest proteomics data set. And so you might ask, what are we doing with this data set? What is it teaching us about genetics, about biomarkers, and about drug targets? Well, let's start with genetics. First of all, you heard a little bit about this already, but we've conducted GWAS for all 3,000 proteins. And overall, we've identified 14,000 genetic associations with proteins. We call these protein quantitative trait loci, or PQTLs for short. And the main takeaway message here is that most of those PQTLs are in non-coding regions. This is just another line of evidence to support the idea that junk DNA was always sort of a junk concept. Most of the variants are in non-coding regions. And about 86% of the variants that we identified were influencing proteins that were encoded far away. So we call these kinds of PQTLs trans PQTLs. And those trans PQTLs are very useful to help disentangle very intricate biological networks. So take, for example, on the screen, the complement pathway. You have your classical pathway for the complement, your lectin pathway, and your alternative pathway. And what we were seeing is, yes, there's lots of cis PQTLs, proteins within, let's say, MASP1 that influence MASP1 protein levels. But more interestingly, there are lots of trans PQTLs showing a complex mix of local and longer range effects. In the case of MASP1, for example, there are variants within C1, within FCN3, within vitronectin. These are all genes that encode faraway components of the complement pathway, and they're all influencing MASP1. There's also genes that are not strictly part of the complement pathway that influence MASP1, such as variants at protein C inhibitor, which is part of a coagulation pathway that's closely tied to the complement pathway. And conversely, there are variants within the MASP1 gene that influence protein levels in other components of the complement pathway, such as factor D. So once again, this is all just by way of saying it's helping us disentangle these complex networks and see how that there are both local and longer range effects. And I won't spend too long on this slide because Dr. Nag and Dr. Cortes already alluded to it a little bit, but we can plug the proteomics into our existing exome-wide association studies and studies of human knockouts, and it actually gives us a stronger signal. And that's pretty intuitive. If you can actually show that your protein truncating variant indeed is associated with reduced protein levels, then it's more likely to truly be a loss of function variant, and you're going to get a stronger signal in your association studies. That's a quick overview of what we're learning about genetics from the proteins, but we're also learning an awful lot about biomarkers. So first of all, when we take these proteins together and run multivariate modeling, as you can see, they do a very good job of predicting age, sex, and BMI. For sex alone, the area under the curve is 0.998. It's almost perfect. And for physiological functions like liver and kidney function, once again, we see a very high predictive validity for EGFR, ALT, and AST. And when we look at the proteins one by one on an individual basis, we also learn some fascinating insights. So my colleague Li Ping Hu and Janssen ran cross-sectional analyses of these proteins across over 1,500 diseases. And so what you're looking at here are volcano plots. Each of the dots represents a disease. You have your effect size on the x-axis and your p-value on the y. And we have two different proteins. One on the left is neurofilament light chain. The one on the right is GFAP. Both of those proteins have been proposed to be markers of brain injury in neurodegenerative illnesses. But what you'll notice is that on the left, indeed, neurofilament is associated with dementias in purple, but it's also associated with a lot of non-neurological conditions. We have chronic renal failure, we have hypertensive heart disease, type 1 diabetes. So it's a little bit of a messy marker. It does give us some sense of brain injury, but it's associated with a lot of other things. That contrasts with GFAP. It's associated with far fewer diseases at the same level of statistical significance, and most of those diseases are neurodegenerative. Most of them are brain illnesses. So this is all by way of saying UK Biobank is helping us to understand which biomarkers are more specific and which ones might be a little messier, a little more pleiotropic. And the last example I'll give on biomarkers is for ischemic heart disease. So as most of us know, NT-proBNP is already typically implemented in clinical practice for diagnostic purposes for heart disease. And indeed, when we run the cross-sectional analyses, it's the top protein. It's the most significantly associated protein with heart disease. But what you'll also notice is that there are other proteins that are at a comparable level of statistical significance. There's renin, galectin-4, NPPB. So this is all by way of saying that in the future, we may want to consider measuring many different proteins at the same time to get a more nuanced picture of our patients' health profiles. And then the final thing that I'll talk about is how the proteomic profiling of UKB is helping us find better drug targets. You've heard about this a little bit already, and I'll give a more unique example to Janssen. Another colleague of mine, Nasser Dousparast, has combines traditional susceptibility GWAS with the protein GWAS to help find new targets for Parkinson's disease. You can see that he ran a traditional susceptibility GWAS for Parkinson's, found a number of the established GWAS loci in LRRK2 and synuclein, etc. And then he asked, of these disease-conferring loci, which of them are PQTLs from the PPP study? And he found that those disease-conferring Parkinson's loci influenced 124 different protein levels. When he looked at those proteins, they were enriched for pathways that are involved in Parkinson's disease, lysosomal pathways, cytokine receptor interactions, etc. He then plugged those proteins into a protein-protein interaction network, found a number of the typical hub proteins like synuclein, but also found a number of new protein hubs. One of them was galectin-3, which is a microglial expressed protein, and galectin-3 has been theorized to be involved in processing of an abnormal form of alpha-synuclein, a toxic form of alpha-synuclein. So this has been published on before in vivo, but this is the first time from a human data set that we've been able to verify its role as a potential target for Parkinson's. And of course, we've conducted Mendelian randomization. Many of you have probably heard about an MR already. I don't have too much time left to explain it in detail, but it's essentially like running a clinical trial using genetic data, protein data, and disease outcomes. And we've conducted Mendelian randomization across all of the proteins in UK Biobank and hundreds of different diseases. I can't talk about some of the new things we're learning due to Janssen legal restrictions, but what I can talk about is PCSK9. It's a well-verified drug target that has been verified by human genetics. We validated its role in hypercholesterolemia using these data. We also show that it may have a causal relationship with ischemic stroke as well. So I'll just finish up by saying that by combining genetic data with proteomics in the UK Biobank, pharmaceutical companies are discovering really powerful new drug targets and biomarkers. And those targets and biomarkers are strongly grounded in human biology. So this should lay a foundation for more of a precision medicine approach in drug development. And as I said at the beginning, the first half of these proteins is now fully available to verified UK Biobank users. And I'd encourage all of you to go take a look at that data. This was a massive scale collaboration, and too many people to thank. I'll just call out Ben Sun from Biogen, who ran many of the genetic analyses you saw today, as well as Melissa Miller from Pfizer and Lyndon Mittenall from Regeneron. Thank you very much.
Fantastic, fantastic, Chris. Okay, so last talk, and Asha Collins from DNA Nexus, cloud-based computing for global collaboration and drug discovery. And then after Asha finishes, I'm going to bring up everybody to the podium, and we're going to have a healthy question and answer session. So Asha, finish us off with cloud-based types of presentation. Off you go.
Absolutely. My name is Asha Collins, and first of all, I just want to thank the organizers for inviting me to speak today about cloud computing and how our cloud-based platform at DNA Nexus is helping to accelerate drug discovery. I'm going to take one step back and just give an overview of sort of DNA Nexus, because I think its history, but also sort of our platform and these key characteristics are really key to some of the things that we're solving with sort of large-scale multi-omics, multimodal cloud computing analysis. So we are a company that was founded in 2009. So we've been around for a decade and a half. We started, as our name suggests, by focusing on DNA, by focusing on genomics. And because we have been around sort of in this industry for that long, really sort of ahead of the curve in a lot of ways, we've learned a lot along the way. And when the partnership and the opportunity to work with UK Biobank came along, it was because of the long history we had with genomics, but then now the multi-omics analysis experience that we have, that I think we're really able to help accelerate some of the goals that UK Biobank had. So who exactly are we? So we are a clinical omics healthcare data platform that really looks to help healthcare and drug development companies accelerate their research. We're cloud-based. We're in several dozen countries. We have tens of thousands of users actually now. And we also have one of the largest databases of genomic data. So we have sort of 70 petabytes of data and growing, and that's been growing every year, 70% year over year. And as we sort of talk about the scale of the UKB data, that will become really important as well. The last two things I'll talk about is one, especially now in this age of really increasing privacy, security, and compliance concerns, we really focus quite a lot on our ability to be leading edge and industry leading in security and compliance. And this is something that we're very proud of. Many of our customers, when they audit our platforms and our environment, actually find that our environment is actually more compliant and more secure than theirs. And because we know this is extremely important for keeping the trust of our customers, but also more importantly, the trust of participants as well as patients, we really make sure that this is something that we are always innovating on. And then last, of course, like most companies, we really pride ourselves on the fact that we have a really great, amazing team that is composed of scientists, bioinformaticians, as well as technologists. And really the combination of all of these types of technologists and scientists really help us understand what is needed to really accelerate drug discovery and drug development in the cloud. And the very last thing I'll say here is that the last two pieces, the footnotes on this slide, is sort of Fast Company and Frost & Sullivan, and it's just really to, again, say we're really excited that we were named the #4 most innovative biotech company this year, and actually by Frost & Sullivan, the #1 precision medicine informatics company in the world. And that's just some background to tell you a little bit more about what our platform actually is. So what do we actually do? So this is a cartoon of our platform, and I'm going to take some time to simply walk us through all the different pieces of this, because I think it's key to really understanding sort of the impact that you can have in cloud computing with large-scale omics and multimodal analysis. So we move from left to right. On the left side, you'll see that you have data inputs. And this is where, again, we have the ability to pull in genomics data, but also proteomics data, as well as metabolomics data, as well as clinical data, as well as imaging data. So we have effectively APIs and feeds where our researchers and our users can pull in any type of data effectively that they would like to into our platform. Once they're in our platform environment, they then have the ability to use the over 200 bioinformatics tools that we have that are out of the box. So we really allow for people to come onto the platform, just bring your data, and you have the analysis tools that you want at your fingers. However, for users that actually want to bring their custom workflows and pipelines into our platform, we also allow for that too. And so that's what we have, so the language bindings. We allow for Jupyter, as well as Spark Jupyter, notebooks, and pipelines we brought onto the platform. And then, of course, we also have a full suite of machine learning and AI tools for users to also accelerate some of their learning and their analysis as well. This is all within a very constrained and actually sort of secure platform or environment that is, again, has the leading security and compliance environment, so in regulation, so we're ISO 27001 NIST 800. Every region across the world we effectively can operate in, and that really allows us to not only just allow for single researchers to be on the platform, but to also allow for global collaborations, which we know are really at the heart of a lot of this large-scale omics research. The last piece I'll say, too, is when you actually look at the right-hand side of this slide, what it also allows for people to do is either use sort of a GUI interface or a command line interface. So if you're a biologist like me, you might prefer to have a user interface. However, if you're a skilled bioinformatician that likes to actually code and really wants to drive customized analysis, we also have a command line interface that allows you to really customize the type of analysis that you're looking to do. And again, because this is actually based in the cloud, and as this depicts, we actually run on AWS as well as Azure. Because it runs in the cloud, that means that the data, the analysis that is being done is a single copy that can be accessed by people wherever they are. So there's never any issue with sort of data versioning. There's never any issue with what data or what type of analysis that people have. It's based in the cloud. It's a single source that all collaborators can touch into, and be able to analyze the data in a very cohesive way. The other thing I'll say about the cloud-based platform that we have is the scalability piece. And this is also really important because if you're working in an HPC environment or local servers, you may have to work in a way that is based on when that might be available. And you also might have to sort of make sure that you're reserving the very specific amount that you need. With the cloud, things are on demand. Things are scalable. If you need more workers, if you need more GPU, if you need to upscale your analysis, if you need to all of a sudden really accelerate it, you have it there for yourself. It's on demand and you can go. And that's something that we see, especially in these large-scale analysis, our customers really leaning into, and we're also really leaning into as well to see what are all the different technological advancements that will allow us to even accelerate that on-demand capability even more. So what does this actually look like when you're actually working in our platform? So the first thing that we actually look at is when you're actually moving into the platform. You and your collaborators are moving to the platform, and we want to make sure that you, as well as your collaborators, as well as other customers, have only the access to the data that they're verified to have. So we ensure that the right people have the right data at the right time, and that people outside of the collaboration circle clearly don't have access to data, or they don't even have access to data within the same collaboration that they're not supposed to have access to. The second thing is we also have what we call a cohort browser analysis. So if you're interested in women over 50 who have been smoking for the last 20 years, we have a tool, cohort browser, that allows you to pull the data from those specific participants and look at them as a cohort and do analysis on that specific cohort and to also visualize it. And there's a depiction of that right here. We also have a much broader sort of suite of ad hoc statistical tools, business intelligence analysis, and reports that will also allow you to take that information, to drive it through different types of tools, and to say, what else can we learn here, and what are we actually seeing across the different cohorts? This, again, can be done either with our built-in tools or with your own customized tools through some of the web applications that we make available on the platform. And last, this can be done not just on a single ad hoc basis, but our platform has enough sort of capability, enough power to actually do this at production level scale. And there are a number of our sort of large pharma customers that really use this to drive their entire drug discovery pipeline. So when we look at this all together, the reason why we are a cloud-first solution and we really lean into the cloud computing is because when we actually look at this, we actually know this actually costs less than an HPC servers and sort of on-premise solutions, not only in the initial investment, but also in the ongoing investment. Again, it really gives the ability to support global collaborations with less bureaucracy, less versioning, less data access issues. It's clear, it's secure, and it allows for the right security as well as privacy. Again, we'll see that there's also just an accelerated turnaround time, the ability to have a distributed cloud network to run your analysis versus on-premise options really allows for people to see what they've done in weeks, go to sort of days or hours, and we've seen this over and over again. Again, part of that is because of the on-demand scalability, and then just last, the thing that is actually first is really around security and compliance. And the picture on the right is just another actual independent analysis that was done that actually looked at some of the benefits, the quantitative benefits that Forrester Research actually saw when looking at this sort of cloud computing solution. Is really time, cost, and quality, as well as security. Those things that really help people get first to publish, first to market, much more quickly. So when we saw the RFP come out from the UK Biobank, looking to have a solution that democratized access to data, we were extremely excited because for all the reasons I just talked about, we thought our solution would be an amazing fit to accelerate and to meet their goals. So what UK Biobank wanted, as has been discussed before, they wanted to ensure that the amazing data that they've collected would be able to be in the hands of every single validated researcher across the world, regardless if they had significant amount of resources or if they were simply just a very talented scientist. They wanted to make sure that the need to have the resource of HPC servers or on-premise capabilities wasn't something they would have to get over to just be able to contribute to science and healthcare innovation. And so what we provided was a platform that is quite similar to our base platform with a few more things, but it's an all-in-one platform that brings the cloud, that brings the tools, and then houses the UK Biobank data. Again, this is secure, so only verified researchers that are verified for specific data can access that data at any point. However, wherever they are sitting in the world, they can access this data without having to have any infrastructure capability themselves. So this is a very simplified version of that wrap architecture. And again, it's very similar to what we have as our core platform, but there's a number of very significant differences. One, when we look at the UK Biobank web services and that double arrow, that's really speaking to the ability for us to verify specific users. So UK researchers apply to UK Biobank and they get approved. Then users come onto our platform and they are then given access to the platform only based on what is provided to us from the UK Biobank web services. And that sort of authentication management system is extremely important, and again, having the right security and privacy for participants' data. The second thing that's not really shown here, but is extremely important, is that we also wanted to make sure that the data of participants was protected as much as possible. And so if there are two researchers, we've put in place a mechanism called pseudonymization, which means that if there are two researchers and they have the same participant's data, they will actually have different identifiers for that participant. So it makes it relatively extremely hard and rare to be able to combine any data sets and to identify participants. We do this at the file level, the file naming level. We also do this at the file content level. And as Naomi showed earlier, we have over 10 million files in the platform for UK Biobank. So you can see this is a significant technological hurdle for us to do. But it was something that was extremely important, and again, can be done because of this sort of cloud background. The second thing that we were also able to do too is simply actually just build out the technology infrastructure with the support of actually AWS, because holding this amount of data, managing, we have over 30 petabytes of data right now, managing 30 petabytes of data from different data providers and also sending it out to, or enabling users to access it from different places around the world is also not something that's insignificant. It was a significant adventure and challenge, I'd say, for our technical team. But that's what we've done, in addition to also simply enabling our entire platform for the global user group of UK Biobank. And what does that look like? I'll keep going, so I think I'm running out of time. Well, first, we launched in September 2021, so we're coming up on our 2-year anniversary. We've grown threefold in the first year. We have now actually over 4,500 users. Over that sort of past year and a half, we've actually had 3 record-breaking data releases on the platform. So we've had the single largest whole genome sequence release ever, the single largest whole exome release ever, and the largest proteomics data release ever on the platform. And I mention this because releasing this amount of data to not just a single institution, but to multiple institutions, hundreds of institutions around the globe at the same time, at this amount and this scale of data is not insignificant, and ensuring that there are no security issues, there are no privacy issues, and everyone has the appropriate access, again, was something that was not trivial, and that we are continuing to actually build on and innovate around, because we're, of course, getting more and more data, and we have more historical releases to do on the platform with our users and with UK Biobank. Again, we have over 17 million files now, over 31 petabytes of data. Since we launched, we've done over 14 different data releases. And here are just a few of the things that we've heard from some of our users. And this is really exciting for us, and you can actually go onto the blog that we have online too and actually read the entire articles that are from our different users here. But effectively what we're hearing back is exactly what I've told you before, but it's always great to hear from people actually using the actual platform. It costs less, it takes less time, it's easier to collaborate, it's more secure. Those are our bread and butter, and that's why we think this the cloud computing environment is so important and helpful to help accelerate really drug discovery, but especially drug discovery at this scale of data. And we do a number of other things as well, in addition to the platform, to simply support the user community. Everything from education and awareness campaigns to user support and success, community engagement. This has been a true honor to be a part of the UK Biobank program, to really support UK Biobank and its users. And we're really excited about everything that we have to look forward to, as well as everything that we've already accomplished together. And at the end of the day, helping our users, customers, and more importantly, helping sort of our healthcare industry. And this is just, if anyone wants a QR code, you can join UK Biobank Wrap, and you can receive 40 free pounds to get started. Thank you.
Okay, can I ask, thanks very much, Asha, excellent. Can I, don't come over here. So can I ask all the speakers to come up? There's enough chairs for everybody. And as you're coming up, again, any questions on any aspect of UK Biobank are welcome. So please come up, tell us who you are, where you're from, and your question, and then we'll just have a healthy question and answer session. And I will probably intersperse with a few questions of my own, with Chairman's prerogative, actually. So make yourself comfortable, everyone. Grab yourself some water if you want some, and we'll have to share microphones between us. So somebody in number one, tell us who you are and where you're from, and your question, please.
Thank you. Saad Shakir from the Drug Safety Research Unit in Southampton. Thank you very much. I enjoyed them.
Sorry, could you speak a bit slower, because the acoustics are a bit difficult up here.
Yeah, I know. I'm hearing my echo. Yeah, I'm Saad Shakir from the Drug Safety Research Unit in Southampton. Thank you very much for the speakers. I have learned, although the last presentation was a sales pitch, which should have been not be a sales pitch, it was quite like that. My plea and question to the UK Biobank is that we were, while applying to do a study on thrombosis and thrombocytopenia with COVID-19 with the biobank, and it occurred in young people who were young 2 or 3 years ago. But your cohorts who were 40 to 60-year-olds when they were recruited makes the population of the UK Biobank now older. So what are your plans to include younger people in the biobank, please?
Naomi, it's for you now. Sorry.
Yeah, thanks. That's a great question. You're absolutely right. The average age now is 69, 70, ranging from kind of mid-50s to mid-80s now. There is a lot of interest, particularly from the genetics community, to expand the recruitment to first-degree relatives, so the children and the siblings of participants, in order to look at direct genetic hereditary effects and also shared environmental effects. So that's one of the things that we would like to do over the next couple of years, is to expand the recruitment to the children.
And who's going to pay for that, and is there money?
Well, that's why I said that's what we'd like to do.
Okay.
It's something we could pilot.
New younger people, not the relatives. Are you planning to recruit new younger people who are now like...
So a younger cohort.
It would, if we were to do this, it would be the children of the participants.
Okay.
Okay.
Yeah.
Thank you.
I think, is that somebody up here for a question?
Yes, first I would like to thank all the speakers for their amazing talks. I have two questions about drug data availability through the UK Biobank. So for hospital prescribing data, especially for anti-cancer drugs, are they available through the UK Biobank platforms? And if not, is there a plan in the future to include them? Because I am working in cancer pharmacogenomics, and I know it's really difficult to get these data from hospitals, especially different hospitals can have different chemocare systems. Also for the...
Right, stop. We could okay. Let's answer that question first. Did you pick that up?
No, sorry, I didn't get the question.
Are you getting hospital-type data into UK Biobank? So someone is admitted, are you getting all of that information?
Yes, it's a short answer. We currently have data available from hospital inpatient admissions, including critical care. What we don't have available yet is data from outpatients or clinics, but we do have hospital inpatient admission data, and that data is all available through the research analysis platform and also separately through the showcase.
Okay. Did you have another question?
Yeah, regarding the...
Very quick.
Okay, regarding the OTC drugs and herbal supplements. So are they included in the information collected from patients during follow-up? Because as you know, they are important for the drug-drug interactions.
Naomi obviously hasn't tuned in. Herbal supplements, are you questioners and supplements?
We ask at recruitment, when participants come along to an assessment centre, about any over-the-counter medications or supplements that they are taking, but we haven't followed that up. So we ask them once at baseline, and again if they attend an imaging centre, through self-reported, but we don't follow that up through linkage to any systems because it's just not simply available. But we do have it self-reported.
Okay,
thank you.
I'm going to ask a few questions and we'll come back to you. I can see people standing up just to spread it out. So Abhishek, you sort of mentioned some analysis and you put up the figure 3.4 billion types of data points and then the p-value of 1 times 10 to the minus 8.
Yeah.
Is there still a potential for false positive substantial?
Yeah, I mean, so this is a question we get asked, and we actually published that in our Nature FEWAS paper, which came out last year. So we don't follow the conventional Bonferroni correction as one does in GWASs, where you just divide 0.05 by the number of independent, LD independent tests that you're performing. Yeah. Here, because we are using many different collapsing models, many of which are highly correlated, because we have some models which have PTVs, some which have PTVs and missense, so they will be correlated. So there is correlation at the level of the models, and there is also a lot of correlation at the level of the phenotypes, because we define union phenotypes which combine data from the different sources. So because of the correlation, what we do is that we run permutation analysis where we permute the phenotype and try to decide that threshold below which we do not see any associations just by chance. And we also use what I did not mention amongst the models is a synonymous model. So we would expect that to be an empirical null under the assumption that synonymous variants within a gene won't associate with phenotypes. And the two approaches that we use coincided very well at that p-value threshold of 1 into 10 to the power minus 8.
That's a complex answer. I only understood some of it, but it doesn't matter. So before, Adrian, I'm going to come to you, then I'll come back to speak to out there. So 2 times for genetics, that genetics have improved things twofold. Is that good enough, or why is it not better than that? Can you maybe get the microphone there? That's a tough question, I know. Or are things improving as we're getting better at genetics and understanding and trying to exclude pleiotropy and various other aspects?
Well, you ask
so right into the...
You asked me, it's good enough. Well, it is bigger than 1, right? So that already gives you a strategy to increase probability of success, right? You saw the graph, right? And you see that increment in probability of success as development progresses. Now, this is all looking retrospectively, like before actually GWAS era, right? So this first study was looking at trials that started before the era of GWAS. So now, I mean, now this actual analysis is kind of hard to perform because now you have all of this wave of genetic insights that are going into
I think things are improving.
Development, right? Yeah. So hopefully we will see that improvement in
I think you're right. I think my sense, even from all the talks, that things are massively improved as the technology, as we've got evidence of proteins, so we've got lots of other information to allow us to be more precise. So I think, young gentleman up there, number 1 or 2, I can't see from here. Go ahead, tell us who you are and please give us your question. Yeah, it's yourself, yep.
Glad you heard. Hi, I'm Jeff Aronson. I'm from the Centre for Evidence-Based Medicine in Oxford. Some years ago, the late Richard Green and I invented the rather clunky term targetophilia to express our concern that people were being too target-driven and neglecting systems pharmacology, and we've heard some of that today. You hit a target, it upregulates, or it goes down a different pathway, and you lose out. The example of psoriasis was great, and migraine and other polygenic and complex diseases. So my question is, should we, like we heard in hypertension yesterday, where they're using 5 different drugs for different targets, should we be thinking about combining target-driven approaches to use multiple drugs for treatment of polygenic complex diseases? Or should we even, in addition to looking for drugs that are specifically target-driven, should we be thinking about looking for drugs that have multiple targets that together might be synergistic or at least additive?
Okay, so I think this is best answered by Mary Helen. So, and I love that talk as well, because we've just put in a grant, Mary Helen, on looking at weight loss in psoriasis and psoriatic arthritis, because obesity is a big causal, it's a causal factor, but that might be, maybe the way to answer it, might they be using the genetics, and precision medicine is a big question, to be able to identify those who will benefit more from one particular type of intervention and those who require other types of intervention.
That's right. So I think
Again, could you speak right fine?
we have many tools for modeling patient stratification that can answer that question, right? So as you saw in the example of migraine, for example, polygenic risk appeared to modulate the efficacy of one class of drugs, but not another, right? And so I think that is an important feature to consider. In regard to combination therapy, again, we have ways to model interactions of functional variants in multiple genes that can answer that question as well. And then if you think about that in the context of stratification, perhaps based on polygenic risk score, right? You could think about precision medicine strategies for, let's say, deploying, bispecifics in particular diseases.
Okay, fantastic. And I guess I'll maybe pick on Chris, and then I'll come back to the questions. Chris, the proteomics is all there, and you mentioned that it's allowed you to perhaps define causality, but proteins themselves are protein measurements. I'm not 100% sure in my mind how good Olink is across the spectrum in terms of how precise the measurement is, but equally, not all proteins are causal. Some of the changes happen as a consequence of something else upstream and some downstream. How do you disentangle that? Is that the MR bit you showed at the very end?
Exactly, yeah, that would be the MR bit. So, to explain it, to oversimplify the explanation, it's like running a clinical trial. We're all, assorted either into genotype A or genotype B, and then you can use your protein or your PQTL a little bit like you would use a biomarker in a clinical trial. So that's, we combine MR with multi-trait co-localization. That's the best method we have at the moment to try to infer causality. I think some of the cross-sectional tests are helping us as well to get a sense of, okay, is a protein causally associated with disease, and then is it active in the disease state as well? Is it active when you're looking at somebody's blood sample with a prevalent disease?
Okay, great. I think number 4 was next, please.
Thank you. My name's Emma Baker. I'm a professor of clinical pharmacology at St George's in London. My question is sort of complicated. My first question is about the proteomics. Presumably this is on samples that were collected 15 years ago at baseline. So it's a snapshot, I guess, 15 years ago. So I'd just like some sort of comment on, have the proteins, could they have degraded or changed in the samples, but also are there repeat samples across the cohort, etc. And then it sort of comes also to the first question about drugs. What drugs are these patients on? Do we know whether their outcomes have been modified by their statins or their diabetes drugs, etc.? So
well, I can take some of that. So the answer in terms of repeat, they have samples to be able to do repeat, probably the 100,000 at imaging. They're still available to be measured, and people need to put in proposals. And if they get enough funding, they will do them again. We'll probably put in a proposal to measure some specific markers. I'm not sure if that will come off. And in terms of stability, I don't know if anyone wants to pick that up. Chris, maybe, more proteins are much more stable than you imagine.
Yeah,
Chris?
No, absolutely, yeah. The proteins look very, very stable based on what we're seeing. We run very stringent QC on the data that we've generated, and I think that only one of those 2,923 proteins actually dropped out based on QC, to the best of my recollection. So very stable. And a lot of the samples, they haven't undergone too many freeze-thaw cycles, etc. So they're very high-quality samples as well.
And the last answer to your point, yes, they do know, because patients were asked what drugs they're on, and so that, that's all known, so you can adjust and correct for those aspects as well. Naomi, do you want to pick up?
Yeah, in addition to the participants self-reporting what medications they're on when they joined the study, we also have primary care data on about half the cohort, which has prescriptions, and we're also just scoping out the feasibility of linking to dispensing data as well.
Yeah, and I think that allows you to try and overcome some of those confounding aspects that you're concerned about. But, as you can see, some of the results that the guys presented, there's still some fantastic results come up. So even though you've got those concerns, the data...
I wasn't actually concerned. I also thought it was a fantastic opportunity to sort of almost confirm what some of our drugs are actually doing.
Yeah, no, fair point. Yeah. I mean, the statins, pleiotropy, I'm not convinced they're pleiotropic, but that's the kind of an example you could potentially test, I'm going to, before I come back to number 3, Asha, I don't think I was a bit unfair about sales pitch. You're allowed to do that. But I want to know how long it took you guys in UK Biobank to actually get that data over to DNA Nexus and get the whole thing up and running. How long did it take?
Yeah, so we said there have been 14 data sets that we've sort of released onto the platform since 2021. Some of the larger ones, especially the whole genome sequence data, they've taken months to get to us and to transfer. And then the other piece of that too is you get the data, then you need to sort of check it. And when you're talking about millions of files, and how do you even just scientifically, mathematically, how many of those do you check? How do you actually check those? What are you checking them for? That's sort of another sort of several weeks to months as well.
And how many staff do you have? Just out of interest?
There are actually 3 people who actually do all of our data operations.
Wow, okay.
Yeah.
Fantastic.
Fantastic.
Right. Number 3, who's been waiting patiently.
Thank you. Actually, I have a question for Asha. So my question is about the DNA Nexus platform. So if you have access, say, to UK Biobank data, do you have to pay extra to use DNA Nexus, or can that access that you paid for through UK Biobank cover that?
I know what the answer is going to be.
You laugh and give it to me. I feel like that's it. So the access fees that you pay through UK Biobank allows you to get access to the UK Biobank RAP platform. This isn't our platform. This is a UK Biobank research analysis platform that we've deployed for them. And once you're on the platform, all of your compute and usage will then get, after you use your free £40, then the rest of the compute and usage will then get billed to you or whichever sort of wallet or account that you set up. This is also where you could also, I'll go to you too. This is also, though, if you're a early career researcher or a researcher from a low and middle income country, there's literally $500,000 sort of set aside to support those types of researchers. So if you are, again, in either of those categories, I would definitely encourage you to apply for those grants.
Before we come back to number 1, Abhishek, I want to ask you, you showed a lovely example of MAP 3K15, if I got it correctly, and you said, did you say that somebody's developing targets to this, or is that happening? But you also said it wasn't linked to BMI, but did you also, could it be linked to body composition rather than just BMI? So have you done those types of analysis as well?
Yeah. So to answer your first question, yes. So it's now in the scientific domain. We have published this paper, so I'm sure people would be, pharmaceutical companies that are interested would be interested in targeting this potentially. Within AZ also we have our internal program, so that follows the normal drug development pipeline. This is a target, so it has to follow the processes. We know there are certain non-specific MAP3 kinase inhibitors out there, so not specifically for MAP3K15. So there's a publication which was, I think, a couple of years back from Gilead around, MAP3K550 non-specific inhibitor, which was used for NASH. So there are compounds out there, to answer your question, that's being actively pursued by us. And second part was?
No, I think that was...
Oh yeah, the BMI part, yeah, we did use some of the body measures, which fat and lean mass measures, which have come from the imaging studies. We found that all independent, actually, the protective effects.
So it sounds like it's not to do with where your fat is, it's maybe more perhaps some other aspects we don't fully understand.
Yeah, potentially, beta cell protection,
yes,
that kind of
I wonder if
hypothesis.
it's and maybe Helen, so, and all of you guys, I mean, precision medicine is said all the time. All the time. Now, you guys give some brilliant examples, the best I've seen, but in some of the examples, still 25% of people who didn't have the additional testing did respond. So can we actually still deny treatment to people if they don't have a particular, phenotype, or how convinced are you precision medicine is going to be as big a thing as some people think it is?
Yeah, that's a great question.
And that maybe come to all of you if you want a quick answer to that.
I'll just jump in.
Yeah, you go ahead.
I think that things like polygenic risk scores and perhaps other, quantitative biomarkers and things can be used to define a profile of individuals who are more likely to respond.
Okay.
That doesn't mean that the people that don't have that profile or at the extremes of that profile will not respond. As you pointed out, there are plenty of responders, even in people, let's say, of lower genetic risk, right? So I don't think we should think about, not treating people if they don't meet that profile. However, when you think about clinical trial design and you think that you want to really sort of maximize, your sample size for lower cost, right? It's really useful to think about enrichment of events,
yes,
right? Okay. And increasing effect size in your trial. And if it turns out that, we have medications that are particularly expensive, right? I mean, you can think of the, GLP right now as an example. If we could identify individuals who are most likely to benefit from those therapies, right, then we might have a better chance, at least in the United States, of convincing payers that this is something that should be more widely applicable.
Great answer. Anybody else want to comment, or I think that's a pretty, Adrian, sorry. Yeah.
Just one minor comment. So, because you just said that you deny the treatment, but I think the aspiration is that you know which treatment to choose from, right? So we had the example of psoriasis and TYK2, but you also have very effective anti-TNF therapies for psoriasis, right? So hopefully, precision medicine would allow you to choose, well, do you give it an anti-TNF or you give it an anti-TYK2?
Okay, yeah.
It's not there yet, right? But
no, you're good point. And do we also think about weight loss for that individual and so on and so on? Okay, I'm going to give the last two questions. I mean, we've got plenty of time, so if there's any other burning questions, please come up. So, number one, please.
Thank you. My question is sort of a follow-up to your question, Mr. Chairman. I just want to get insight, how you think and develop your strategy. Because a situation where you may have one or more genetic causes, one or more protein causes with variation in the certainty, and one or several environmental factors with various degrees of certainties about each of them. When you put all in the mix, how in the drug development, decide what is the strategy? Or are you going to do, a 2-arm study, a 3-arm study, a 5-arm study? I just want to understand how you deal with the uncertainties of these combinations, especially in the drug development, and especially early in the drug development when you're choosing targets or systems.
Yeah, that's a tough question. I don't know. I'm not sure if does anybody want to pick that up, or can we leave that for maybe a conversation offline? It's a very important question. I guess my sense is you guys are developing all the data, and it's for other people to pick up and design those trials based on the new information. Anybody want to add anything? No, I don't think so. It's a very tough question, but it's a very good question, and I think it's something that people are going to have to grapple with, all these new bits of information.
Well, we need huge data to answer, because it's multifactorial.
Yeah, no, absolutely. And when I see patients in my clinic, for example, where I've got somebody with NASH, how much of that NASH is genetically driven because they've got propensity towards putting fat in the liver? How much is it alcohol? How much is it obesity? And I guess some of the genetics will allow us to try and work that out, but I don't know. Yeah.
Maybe I can just add something
go ahead, Abhishek,
related to this, and this is something that we get asked when you're actually doing rare variant analysis of common diseases, is that, are you going to target that medication only to the patient subset that is carrying that rare variant? Because then the application might be quite small because they are so rare. So one answer to that tends to be, and I think which is seen with PCSK9 and hypercholesteremia, is that though that rare variant might be carried by a very small subset of the population, it tells you something about the biology, and you can potentially target, if that biology is significant, you can target it for the patients at large. Obviously, there will again be a precision medicine angle where some would respond, more likely to respond, but that's the hope and aim of these rare variant analyses.
Okay, and of course, in all of this, as they go forward, it's going to be cost-effective analysis. How much effort and cost to define a particular phenotype or genotype as against the benefits, so that's going to happen over the next 10, 20, 30 years, yeah. So we're going to give the last question, at number 3, and then we'll, sorry, there was a question there, but okay, one short question, 2 last short questions, number 3, and then right at the beginning. Go ahead.
Thanks. Well, I think the answer may be long, but you guys are all, by definition, enthusiasts. You've given us great talks. But in any system, there are bound to be problems and deficiencies. We've already heard about the lack of young people in biobank, and that's an important deficiency to remedy. Are there any other problems and deficiencies that you people perceive in the biobank that need to be tackled, either remedied or at least ameliorated?
So that's for Naomi, I guess. I mean, how much money could you give Naomi to do many other things? Go ahead.
Yeah, I have a limitless pocket.
Well, apart from finances, one disadvantage of UK Biobank, which is picked up a lot, is the ethnic diversity. So only about 5% of the cohort are black or ethnic minority, which actually was representative of the UK at the time of recruitment, but isn't now. So rather than us trying to recruit or oversample participants from black and ethnic minorities now, well, we're going to have to wait another 15 years to find out what their health outcomes are. I think the better strategy is to set up similar studies like UK Biobank in other populations around the world, whether that's Mexico, whether that's Asia, whether that's Africa or India. So those communities can generate polygenic risk scores, and they have very different environmental and lifestyle factors as well. And that, I think, is a better strategy rather than trying to do it all in the UK.
Yeah, I completely agree. And that would also spread more funding to other parts of the world which desperately need them. So listen, we're going to finish off. Last question, just young gentleman here. Please again tell us who you are and where you're from and your question.
Yeah, Grant Drummond from La Trobe University in Australia. Do you have or do you plan to collect gut microbiome data for your participants?
Did you say microbiome?
Gut microbiome. Yeah.
Wow, it's my favourite topic. Well, it's not actually my favourite topic, but Naomi can tell.
Yeah, well, to be honest, we really wanted to, but we couldn't get the funding.
Yeah.
So, and it seemed, I don't know what your thoughts are on this, but 5 years ago it seemed to be all in the fashion, and now it seems to kind of gone off the boil a little bit. There's lots of variation in how you collect the sample, how you store it, what techniques do you use, and so on. So the short answer is we don't have plans at the moment, no.
I mean, there was a lot of pilot work done to try and look at all of that, and unfortunately it just didn't convince the funders sufficiently. But nevertheless, there's tons of other data, as you can see, that, and I think the beauty of UK Biobank, huge amounts of data, the real key factor that people need to think about is what is the question, and ask questions of it which are worthy and important, and I think that is the absolute key. And I suppose I've got one last question for all of you, is that there's so much data. What kind of researchers do we need in the future? Do we need people who are comfortable with genetics and proteomics and clinical work, or are you finding the way you're working together with people with different expertise able to cross-talk quite well enough? Is that your experience? Is that a general yes? Okay. Okay, well, listen, I think I've tied them enough. Listen, we've done well. We're almost at the hour. 5 minutes to go. I will stop there. I want to thank the panel. It was a brilliant session. Thank you for listening, and enjoy the rest of your day. Thank you.